Assalamu alaikum. Welcome to F6 online support program. This is subject biology, page 97 and 98, unit 15 inheritance, and topic is genotype and its types. Objectives. So by the end of the lesson, you will be able to define genotypes and phenotypes, dominant and recessive alleles, and discuss Mendel laws of inheritance. So starting with the first topic, that is a genotype and its type. So before starting, you should know the actual meaning of the genotype. Geno is basically a term used for the gene and type is for type or form. So we will define the genotype as the specific combination of genes in an individual. This is known as the genotype. So what is meant by this specific combination of genes? Specific combination of genes means that the gene has different types of combinations. It means that the genes exist in pair. So always remember that the gene have specific combination because the gene exists in pair for each characteristics. For example, eye color, hair color, and skin color, each trait or characteristic has its own pair of gene. And these pair of genes are present in the different forms. Secondly, these pairs are present on the homologous chromosomes. So now we can say that the gene have different alternate forms. And these alternate forms are present on the homologous chromosome. So here we came towards the definition of the allele, that is the alternate form of the gene is called the alleles. As a whole, this pair is known as a genotype, but individually it is known as allele. Like for example, this capital A, small a, these are the alternate or these are allele of one another, but they are present on the homologous chromosomes. Now we have two types of the genotype. The one is homozygous genotype and the second is heterozygous genotype. So homo means same and zygous mean zygote. The hetero mean different and zygous is used for the zygote. So homozygous mean the genotype in which the gene pair consists of two identical alleles. That you see here, that is capital A and small a. This one is homozygous dominant genotype or alleles, and this one is the homozygous recessive alleles. Now heterozygous genotype mean the genotype in which the gene pair consists of two different alleles. That you see here that the one is capital A and the second one is small a. Now we have some other terminologies, that is the dominant alleles and recessive alleles. So what does it mean? The dominant allele, when allele express itself represented by the capital letter, and the recessive are those alleles which do not express itself represented by the small letters. So the dominant allele will always dominate itself, will mask the expression of the recessive alleles. So in homozygous genotype, you see here that this is homozygous dominant gene pair and this is homozygous recessive gene pair. But in heterozygous genotype, this is present in heterozygous condition in two different forms, that is capital A and small a. But still here the capital A will always be dominant and it will mask the expression of this small a, or it will not dominate its expression. So the example given in book is of albinism. Albinism is a genetic condition, genetic disorder, or the genetic disease in which the normal body pigments are absent. In a normal human being, there are some pigments present which is responsible for the eye color, skin and hair color. And this pigment is known as melanin. So always remember that the melanin pigment is responsible for the eye, skin and hair color. But the person having albinism has no melanin production and he or she has no skin, eye or hair color. In that condition, the person is totally, completely white. His, her hair, skin, eye color, etc. do not have any color. Albinism is basically a recessive trait and it is produced when both alleles are in recessive condition. In normal human body, the allele, the capital A, the capital gene pair is responsible for the normal body pigment production, that is of melanin, while the small a does not produce any type of the pigment. So in case of homozygous dominant condition and heterozygous condition, the individual will produce the normal pigments. Why we mention heterozygous condition here? Because in heterozygous condition, we have one dominant gene, that is the capital A, which produce the pigment. So in heterozygous condition, the individual body will produce a normal melanin pigment. But in case if the genotype is homozygous recessive, then no pigment will produce and the individual will then become albino, or he or she get the disorder known as the albinism. So now the expression of genotype in the form of trait is known as the phenotype. So we came with the final definition, that is the phenotype, which is the expression of the genotype in the form of trait. It means the physical appearance of an individual is known as his phenotype, which is actually due to the expression of the genotype or the expressions of the genes. Mendel laws of inheritance. Mendel was a monk and he lives in Austria. Mendel developed fundamental principles of the genetics. He studied and worked on pea plant and he selected pea plant to carry out a number of experiments. But the question arises, why he selected the pea plant? So Mendel gave the reason for this question and for this selection. He also discussed that the organisms for the genetic experiment should have the following features. There should be a number of different traits. It means the organism or the plant should have a number of different characteristics, like a pea plant or other experimental plants should have seed color, seed shape, flower position, stem length. These all are different types of characteristics that is present only in pea plant. Organisms should have contrasting traits. Like for example, like we say in pea plant, the seed shape. So in pea plant, the seed shape is round as well as wrinkled. The seed color is yellow as well as green. So there are different contrasting traits present in the pea plant. Organisms should be self-fertilizing, but the cross-fertilizing should also be possible. An organism should have short but fast life cycle. So the pea plant was having a short but fast life cycle. All the features are present in the pea plant. That's why Mendel selected the pea plant. Any other plant can also be selected, but that plant should then have all these characteristics. As you see, these are the different traits present in the pea plant, like seed shape, seed color, flower color, pod shape, pod color, flower position and stem length. So all these are 7 traits that are present in the pea plant, and all these are the contrasting traits. Like seed shape is round as well as wrinkled. Seed color is yellow as well as green. Flower color is purple as well as white. So these are the contrasting traits. Mendel law of segregation. Before starting this law, first we have to understand some terms. That is true breeding, which mean homozygous. It means that the parent have same allele in gene pair. For example, capital A, that is homozygous dominant. Small a, that is the homozygous recessive gene pair. Monohybrid cross, the cross in which we study only one trait at a time, is known as monohybrid cross. Here we will discuss the seed shape as an example of the monohybrid cross. So we have to start with the true breeding. It means we have to select the parents with the true breeding having both dominant and recessive alleles. Here you can see the Mendel crossed true breeding round seed plants with the true breeding wrinkled seed plants. So as in parental generation, you can see that the one parent is true breeding round shape and the second is true breeding wrinkle shaped. So first we form the gametes which are haploid, and the fusion of gametes results in zygote in new individual or F1 generation, which is also called filial generation, and all the resulting individual are round. Why round? Because we have a dominant allele that is of the round shape. So, by self-fertilizing the F1 generation, we get F2 generation, and in the result of F2 generation or in the result of the self-fertilization, we get 75% round shaped seeds and 25% wrinkle shaped seeds. If we take out the ratio, so we have 3% round shaped seeds and only 1% wrinkle shaped seeds. Now coming towards the Mendel law of segregation, which says that during gamete formation, the gene of each pair segregate from each other and each gamete receive one gene from pair. When gametes of the male and female parent unite, the resulting offspring again get gene in pair. It means that when gamete form, allele of the gene get segregate or separate. When the gamete unite, the resulting zygote get the gene again in pair. So this was all about the Mendel law of segregation. The second law is Mendel law of independent assortment. It is also known as a dihybrid cross because here the Mendel studied two contrasting traits at a time. So that's why this is known as a dihybrid cross. He performed experiment on two traits, that is seed shape and seed color. So the round seeds and wrinkle seeds, while yellow color and green color, these are the two traits. So always remember that round seed is dominant over the wrinkle shaped seed, while the yellow color is dominant over the green color in case of the pea plant. Round is controlled by the capital R, while the wrinkle is controlled by the allele small r. Yellow color is controlled by the allele capital Y, which is dominant over the green color controlled by the allele small y. Here you can see the shapes. This is round shaped seed, and the second one is wrinkle shaped seed. The round is dominant while the wrinkle is recessive. So Mendel crossed two breeding plants that had round yellow seeds with wrinkle green seeds. True breeding means homozygous. So round is dominant, yellow is also dominant, while wrinkle and green are both homozygous recessive. So the parental generation consists of one homozygous dominant parent, which is round and yellow, and the second parent is homozygous recessive, that is wrinkle and green. So after getting the gametes and the fusion of the gametes result in first F1 generation, and the F1 generation consists of all round and yellow color seeds. Why round and yellow? Because round is dominant and yellow is also dominant over the green. So the F1 generation consists of all round and yellow color seeds. All the F1 seeds grew into plants and they were then self-fertilized, and which results in 4 different phenotypes. So the F1 was self-fertilized with the F1 generation. After getting the gametes and fusion of the gametes, we get the F2 generation or the second filial generation. Punnett square is used to represent all these crosses. Punnett square is actually a diagram that is used to predict an outcome of the particular cross or breeding experiment. So by this cross we get 4 different types of phenotypes. So the self-fertilization of F1 seeds results in 4 phenotypes, that is round yellow, round green, wrinkle yellow and wrinkle green, and the ratio of these phenotypes is 9:3:3:1. So coming towards the law, it says that the allele of the gene pair segregate independently from the allele of the other gene pair. It means that when individuals differing in 2 or more than 2 pairs of contrasting traits are crossed, so the inheritance of one pair is not affected by the presence of the other pair. Each gene pair will assort independently of the other.